All right, welcome everyone to the session. During the session, we'll be discussing how to annotate epigenetic variation with our package Cocoa. And so if you have any questions, please put your questions in the chat box on Pathable. And I went ahead and recorded a video of the workshop just so nothing would go wrong. So I'll go ahead and share that video, but I'll be watching the chat so I can respond to your questions. All right, let's get started. So a major goal of epigenetic analysis is to understand variation between individuals in gene regulation. A common way of doing this is to have two groups and do differential analysis. So you could compare healthy to disease. Another way you could do this is to look at variation along with a continuous phenotype. For instance, patient survival, cancer stage, age, etc. Or if you don't know the sample's phenotypes, you can do an unsupervised analysis based only on the data itself. So the goal of our method is we want to have a method that could accommodate continuous variation, that doesn't require known groups, so you can do unsupervised analysis, and that leverages covariation of signal and co-regulated loci effectively. And so I'll explain that more later. In this method, our method Cocoa works for data that has values associated with genomic coordinates, such as DNA methylation, ATAC-seq, any similar data. So now I'll just explain the concepts of the method and the background philosophy. So our method is based on this observation that when you have regions in the genome, so there's a little rectangle on here, represent regions, that have shared regulation. So the epigenetic signal will be similar in these regions. So we observed that when you have variation in regulatory signal that controls these regions, then you would also have covariation of these regions across samples. And so using that idea, we can actually work backwards. If we see that there is covariation of epigenetic signal in co-regulated regions, then we can infer that there might be also variation of the regulatory signal controlling those regions. And so our method can be used in both supervised and unsupervised analysis. In unsupervised analysis, we only use the data itself. So we have an epigenetic data matrix that has rows being our regions, our loci in the genome, and columns being samples with the epigenetic signal at each region. And so we do some kind of dimensional reduction, for instance, principal component analysis, and we take the latent factors, the principal components, as our target variable for the analysis. So we want to understand basically what is the meaning, the biological meaning of the epigenetic variation represented by these principal components. In a supervised analysis, we have some kind of phenotype that's known, and we're going to use that as our target variable to see what the epigenetic variation is related to that phenotype and what the biological meaning of that could be. Our next step is to quantify the variation. So we want to see how the variation in the epigenetic data relates to the variation in the target variable. So we can use one of several metrics, such as correlation, to see how the signal in each region in the genome correlates with our target variable. And based on that, we give each locus a score. So just a little aside here. I mentioned that the goal of this method is to basically annotate epigenetic variation. And so the way that we're going to do that is using what are called region sets. A region set is a set of genomic regions that share a biological annotation. And our assumption is that these regions have related regulatory control. This could be transcription factor binding regions, histone modification regions, or open chromatin regions in some cell type or disease state. And there's a similar concept to gene sets for gene expression analysis. And so the idea is, if we see epigenetic variation in a region set, that it can imply regulatory variation of that regulatory pathway or module.
So just a quick aside, we often get, these are often ChIP-seq data sets, such as the transcription factor binding and the histone modification region sets. And we can actually find a lot of those publicly available on Geo or similar sources. So that's something we're going to cover later in the lesson, just how you can find those, how you can get access to publicly available region sets to use for your analysis.
And so back to COGA. We have a database of these region sets. And so in our case, we'll use thousands of these, but we recommend using a large database, which you can find publicly available. We provide access to a database from public sources, or you can find wherever. So basically, we will annotate the variation in data with these region sets. And how we do that is we take the average of all the locus scores that overlap with a given region set to get that region set's score. Once we do that for all the region sets in our database, we can create a ranked list of region sets that represents, it basically tells us which region sets are most associated with the epigenetic variation in our data. And so let me just go through an example of how to apply this, and hopefully it'll make more sense. So we looked at ATAC-seq data for breast cancer patients from the Cancer Genome Atlas. So we have here 73 breast cancer patients, and we're doing an unsupervised analysis using principal component analysis. We have a database of 2,200 region sets, and we want to see what region sets are associated with the principal components of the ATAC-seq data. And hopefully this can shed some light on the biology of the chromatin accessibility variation between samples. And so after we do PCA of the ATAC-seq data, we use the principal components as our target variables and run them through COCO. And so we can see here, I'm showing each region set ranked, and then I'm showing its score. So you can see for principal component 1, the top region sets are estrogen receptor or estrogen receptor related. And so that makes a lot of sense because estrogen receptor is known to be a major prognostic factor in breast cancer. So we can see that there's a lot of chromatin accessibility variation across samples in these estrogen receptor regions, especially along principal component 1. Looking at principal component 2 now, we can see that many of our top region sets are hematopoietic transcription factors. And so we think that this might be related to some kind of variation in the immune component of the tumors. Now for principal component 3, we see that there's not really variation related to either of these things, either the hematopoietic transcription factors or the estrogen receptor. And that'll make sense in a moment. And then principal component 4, we see is similar to principal component 2. And so now looking at the PCA plot, we can see that principal component 1 separates ER positive from ER negative samples pretty well. And that's statistically significant based on Wilcox and Rankstein tests. And so actually, if we look at the p-values for whether the principal component scores are associated with estrogen receptor status, we can see that each of the top 4 principal components, except for PC3 is associated with ER status. And so if you remember, we could find kind of plausible biological interpretations for all the principal components except for PC3, which is the one that's not associated with estrogen receptor status. But the other ones seem to have some kind of meaning that's related to estrogen receptor. And so we can now take our principal components and learn a biological interpretation without even needing the known estrogen receptor status. And so if we look at the raw data in our top region sets, we can see that it basically shows how our method's working. So in these heat maps, we have the rows are samples, and they're ordered by their principal component scores. The columns are regions in our top region sets. And so you can see for estrogen receptor, ER, that along principal component 1, as you go down the heat map, there is huge variation in chromatin accessibility. And so at the top, you can see the ER negative samples, which have low chromatin accessibility in the estrogen receptor binding regions. And at the bottom, you can see where there were the ER positive samples that have high chromatin accessibility. And so basically, our method is identifying these regions that covary across patients along the target variable of interest. And so if you look at sep-P alpha and ERG, which are two hematopoietic transcription factors, you can see that for PC2, there's variation along the principal component 2 axis. It's not as extreme as estrogen receptor, but it still shows why our method was able to pick this up as being associated with principal component 2. And so one other question you might ask is the variation we see, is it specifically related to these region sets? Or is it maybe just across the whole genome or the broader genome? And so we can create these plots called meta-region profiles. And so what we do to create these plots, we take the region set, so for example, estrogen receptor, and we look at a wide region around that region set. So in the center is our 500 base pairs of the estrogen receptor ChIP-seq regions. And we've expanded that to be 14 kb centered on those ChIP-seq binding regions of estrogen receptor. And then we're plotting the correlation between the ATAC-seq signal and the principal component scores. So you can see that a sample's PC1 score correlates with estrogen receptor, or correlates, with chromatin accessibility, specifically at the estrogen receptor binding regions and not at the surrounding genome. And we can see the same thing for GATA3, which is known to associate with estrogen receptor. And so likewise, we see that these hematopoietic transcription factors, the chromatin accessibility is not correlated with principal component 1 score, so we don't see a peak in the middle, but it is correlated with principal component 2 score. And so that basically supports our findings that principal component 2 is associated with immune chromatin variation. All right, so now that we covered this, let me just pause briefly so that we can take any questions that you might have about this presentation.
Yeah, so I haven't seen any questions in the chat, but feel free to ask them now or continue to put them in the chat whenever you want to.
There are a couple of questions in the chat, one in the chat and one in the polls.
Okay, let me take a look.
So there's a question on how you're scoring the enrichment in the principal components, and then why you used only 73 breast cancer patients from TCGA, and what filter you use to reduce from all the more than 1,000 to down to 73.
Sure. So the first question, to get our region set score, it's very simple actually. We are essentially just taking the average of all the individual region scores that overlap with our region set of interest. So that would be, we have a set of the peaks from the chromatin accessibility experiment. And so whichever of those overlap with our region set of interest, we're gonna average those. And there's also some other scoring methods. And we take into account what proportion of the peak region overlaps with our region set region. And then the score that we're averaging is the correlation of the signal in that region with the target variable. So to the second question, there's a lot more data for DNA methylation and RNA-seq, et cetera, for the breast cancer patients. But as far as I'm aware, there's only 70-something ATAC-seq samples for the breast cancer. They could have added more, but that was all we had essentially. So we essentially used just about all of the samples. All right, so I think we'll go ahead and keep moving if there's no other questions.
There's one more in the chat.
Okay.
How do you determine the region set for different transcription factors?
Yeah, okay, let's see. So there's multiple ways you can get a region set. The most common way that we use is to get publicly available ChIP-seq data from various cell lines or tissues. And so it's not perfect because it might not come from the tissue that you're interested in. But a lot of the time it is possible to find a similar tissue or cell type. But it's also possible to define your region sets however you want. So you could use motifs, for instance, and define region sets that way. So it's very flexible. All right, any other questions?
Not that I can see. Oh, there's one more. If in the polls, is there a way to identify epigenetic regulation using bulk RNA-seq if you don't have ATAC-seq data?
There might be, but I think that's beyond the scope of what we're doing here. For this analysis, you have to have some kind of epigenetic data that has regions and signal there. Yes, I think I'm not actually seeing the questions in the chat. So Charlotte, whenever those come up, if you could let me know. Thanks.
Okay, sure. They're in the pathable page in the.
Yeah, for some reason it's not showing those.
Okay, cool.
All right, so now that we've given you an overview of COCO, we can jump into the vignette. And so you have access to this vignette on the web. And so we're going to skip the first 4 sections since we basically covered that in the presentation. Feel free to review that later. But if you click on the COCO for DNA methylation data, that's where we're going to start. And I'll be doing it actually in the console, so I can do it with you. So I'm going to switch over to RStudio now. All right, so our first example will be related to what I showed you in the presentation. We're going to look at DNA methylation data for breast cancer patients from the Cancer Genome Atlas. And so we're going to do both the supervised and unsupervised analysis. And so this time we'll start with supervised. But let me first go over the data with you. So we have 4 different region sets. These would be our region set database. When we did the real analysis, we used over 2,200, but for the vignette, we're just going to use 4. So these were 2 of the top region sets from our analysis, estrogen receptor and GATA3, and 2 of the bottom or least worst scoring region sets from our analysis. And so these are G-ranges objects that have basically the coordinates for those which are ChIP-seq protein binding regions for those transcription factors. And we're only going to look at chromosome 1 in this vignette. So now we're going to load the DNA methylation data. So the coordinates and the methylation proportion from 0 to 1 at those coordinates for each sample. We also want to load the metadata because we're going to use that to do supervised analysis. That's where we have the information whether each sample is ER positive or ER negative. And so let's go ahead and take that information out of the metadata because that's going to be our target variable for this analysis. So now we're just going to convert that information, ER positive or ER negative, into a number. That way we can correlate it with our DNA methylation data. And so you can use multiple different correlation methods. The main thing is that it needs to give a higher value to those CPGs where the DNA methylation correlates with estrogen receptor status. So in this case, we're just going to use simple Pearson correlation. And then we'll just make sure that there weren't any NAs for missing data. Okay, so now if you remember the presentation, we assigned a score to each locus, our feature contribution score, which in this case is the correlation coefficient. And so we're going to aggregate those individual locus scores to score each region set in our database. And so that's what we'll do now. In this case, we're going to take the absolute value, which would allow the DNA methylation to be positively correlated or negatively correlated with estrogen receptor status. So in this case, we don't really care. Okay, so now we can see we have a score for each region set. You can see the final column is region set name. And so we've got the ER status column gives a score. And you can see that the ER and the GATA3 region sets have much higher scores, much higher average correlation than ATF3 and NRF1. And then the function also gives some generic information about these region sets. So signal coverage means that 440, for instance, 440 CPGs were covered by the estrogen receptor region set, whereas 216 of the ER ChIP-seq regions were covered by any data. And so that can just be helpful to gauge how good the coverage was. Generally, we don't want to use anything with coverage less than 100. It's just a rule of thumb, because at that point, the data can get kind of noisy. All right, and then we also have a way to estimate statistical significance. So I didn't mention this in the presentation, but we can actually use a permutation test to assign p-values to the region sets. And so this isn't really necessary when you're doing just an exploratory analysis. Generally, you can just run COCO without the permutation test and get some interesting results. But if you need some p-values for publication, etc., then you can run this permutation test, which is kind of time-consuming. But essentially, we're just going to create null distributions for each region set. And then based on that, we're going to see how extreme our actual scores are compared to these null distributions. And so in this case, just for this example, we're only going to do 5 permutations. But in a real analysis, you should do many more permutations. So hundreds, even thousands. And while in terms of permutations, that might not also be that many, we have a method in our package to use a small number of permutations, so hundreds or 1,000, to estimate p-values. So you can read more about that. There are some caveats with that method. You can read more about that in our preprint of COCO. All right, so here you can see basically the output from the permutation function. What it did was it shuffled the estrogen receptor sample labels, and then it recorrelated the DNA methylation data with the sample labels. And based on that, we could run COCO to get these random permuted scores. And so we can use those scores to make the null distributions. So here we're going to convert those 3 times that we ran COCO into a null distribution for each region set. And so now you can see that, so actually we did 5 permutations. So you can see that there's 5 scores for each region set. And those are the null distribution. So now that we have those 4 null distributions, we are going to approximate the p-values with a gamma distribution. So that's just a method, as I mentioned, to not have to do as many permutations. Okay, so you can see here that we have our column ER status. Basically, the name of that column will be whatever the name of your target variable is. And so that has the p-values for each region set. And so in this case, they aren't really meaningful because we only did 5 permutations. But in the real analysis, estrogen receptor and GATA3 had very significant p-values, whereas NRF1 and ATF3 had much higher p-values. We're going to move on to unsupervised COCO. And this will be very similar. But in this case, imagine that we didn't have the sample metadata about ER status. What if we just wanted to do an unsupervised analysis, do PCA, and then try to understand what the variation was? So we've already got this data loaded, but now we're just going to do a simple PCA of the DNA methylation data. And so let's take a look at the PCA plot. Okay, so here we have just a simple PCA plot, each point being a sample. And we don't necessarily see obvious clusters. Maybe there's a cluster on the left and maybe a cluster on the right, but it's not completely clear. Okay, so in this case, it's an unsupervised analysis, so we're going to treat our principal component scores as our target variables. And we're just going to look at PCs 1 through 4. So once we have those scores for each sample, we can correlate those with the DNA methylation data. And so for those of you familiar with PCA, the correlation of the CPG with the principal component score is very similar to the loadings. In some cases, it's identical to the loadings of the PCA. But if you want to read more about why we're doing this way, you can look at the paper. And so just like our other example, now we have a score for each CPG that represents how much is associated with our target variables. And so now we can go ahead and use our function to aggregate those individual locus scores and score the region sets. And so we see in this case something very similar to what we saw previously. We see that estrogen receptor and GATA3 have really high scores, so really high correlation, and NRF1 and ATF3 have very low average correlation. And so when we, basically the interpretation we would have is that, well, it looks like the top principal components are associated with estrogen receptor with DNA methylation variation in these estrogen receptor binding regions, which would make sense since that's such a big prognostic factor in breast cancer. And so let's just go ahead and look at the plot with known annotation. So this information wasn't used by Coco, but if we plot it here on our PCA plot, we can see that the first principal component is separating ER positive from ER negative patients. And so that's why we get such a high correlation in the ER binding regions between the PC scores and the DNA methylation. And so we have some helpful visualization functions to further understand the results. So the first one is basically a way of understanding just the overall results that you're getting back. So each column we can see that it's ranked the region sets. And so we decided in this case to order by our principal component 1 rankings. And so here on the right, you can see the region set names, and they're ordered by the principal component 1 ranking. And so that's why the PC1 column goes from dark red down to dark blue. But in the other cases, you can see that for PC2, estrogen receptor was actually the second ranked region set. And so basically that's just a way to see whether the region sets that are high for one principal component are also high for the other principal components. And you can also use this for supervised analysis. Okay, so just like with the supervised analysis, we can go ahead and do permutations. And we can basically see similar results. Okay, so there's some ways we have to further understand these results. And so one of those is these meta-region profiles, which are the plots I showed you in the presentation. And so essentially we are just looking at our regions of interest, and then we're going to use the resize function to expand those regions to be 14 kb, but centered on the ChIP-seq binding region. Once we've done that, we can use this function get meta region profile to aggregate the feature contribution scores, which are our correlation values, into a meta region profile. And so there's several parameters such as bin number, etc. Basically, we're aggregating within bins to account for the sparse data. Next, we're going to do some normalization so that we can compare these meta-region profiles from the different region sets and principal components. So here we're just taking the average feature score for each principal component and then subtracting that from the profiles. After that, we're just getting some common scaling parameters for the plots so that we can see them on the same scale. All right, let's take a look at the plots. And so here you don't need to worry about this section. Basically, we're just looping through the plots one by one. As I showed in the presentation, we have these meta-region profiles, and you can see the center is where the ChIP-seq binding region was. And we expanded to 14 kb around that region, around all those regions. And so here you can see the estrogen receptor has a peak in the center around the binding site. So that just means that the DNA methylation, specifically around the binding site, is correlated with estrogen receptor, is correlated with principal component 1 score. Whereas you can look at principal component 4, and we don't see a peak. So that means that DNA methylation is not correlated with principal component 4 scores. We see something similar for GATA3, which is involved in estrogen receptor regulation. We see a pretty distinct peak there. And we see absence of peaks for NRF1 and ATF3. So they actually have lower than average correlation, maybe a little bit, around the binding sites. And so that can basically support your findings and show the specificity of the correlation to your region set of interest and not the surrounding genome. So one other thing we can do is to look at the raw data in our regions of interest. So those would be our estrogen receptor, GATA3, etc. And so you can see here, we input a region set of interest, estrogen receptor, and we plotted the top 100 variables. So these are the 100 variables that are most correlated with DNA methylation level and are also overlapping our region set of interest. And so we've chosen our order by column as PC1, which means that we are ordering our samples, our rows, by the principal component 1 score. And so you can see that along principal component 1, there is DNA methylation variation in the estrogen receptor binding regions. And so now let's take a look at NRF1 and see how it compares. And so in this case, NRF1 had very low scores, it had a very low COCO score for principal component 1. So we don't expect there to really be much DNA methylation variation along principal component 1. And so these are the 100 CPGs that have the highest correlation, but we can see that even selecting the best CPGs, there's hardly any correlation along the principal component 1 axis in this region set, which is why this region set gets a low COCO score for principal component 1. Something else we might want to do is to look at the individual regions in our region set and to see how much they're contributing to the principal component. And also we can use this method to compare to see if some regions in our region set contribute to, say, principal component 1, but not principal component 2, etc. And so here we're showing the percentile of our feature scores, which were the correlation values for the CPGs for each region. And so our rows are regions in the estrogen receptor region set. And so we can see the bright yellow that a lot of these regions had very high percentile of correlations with principal component 1. And it appears that some of the regions are actually important for multiple principal components. Okay, next we'll be moving on to the ATAC-seq portion of this vignette. And so really this will be a lot of review on what we've just covered, because the same functions are being used and the same general method is being used, just with a different type of data. And so in this case, we're still looking at breast cancer, but now we're looking at the chromatin accessibility from an ATAC-seq experiment for breast cancer patients from the Cancer Genome Atlas. And so I'll go a lot faster through this part, since so much of it is review. But I'll just try to emphasize the differences from DNA methylation data.
John, can we make a quick break and answer a question that had to do with the last heat map? So there was a question whether in that last heat map, if the values for each row sum to 1 across the columns.
They do not. So each column is calculated separately. basically we have the average correlation of a given region with the principal component. And then based on the distribution of the correlations for all the CPGs, we can convert that to a percentile. But that calculation is, yeah, separate for each principal component.
Okay.
Okay.
Great.
So here I'm going to load in the ATAC data. And so one difference that I do want to emphasize is that when we're looking at DNA methylation data, we were looking at single nucleotide resolution data. So we had epigenetic signal at a single base, a single CPG. Well, C at least. So in this case, we're looking at the epigenetic signal in regions. And so our data is actually a data matrix where we have the rows are peaks that have already been called, and the columns are samples. And so we want, basically I'm just going to illustrate, I'm just going to show you what it looks like. But basically they've already gone through this and they've called peaks for the samples, and taking their set of consensus peaks, they've then taken the average chromatin accessibility signal in each of those consensus peaks. And so that's basically how we got this matrix. And so you can see that it just has basically the rows are regions, the columns being the samples. And then we've got the chromatin accessibility signal, which has already been normalized as entries. And so the COCO algorithm is very similar to what it was before. And so we're just going to first get out estrogen receptor status for the samples that we're using now. All right, so now let's do another example of a supervised analysis with estrogen receptor status as our target variable. So the main difference here is that there are some additional scoring methods that are available for region-based data. Basically, we can take into account the degree of overlap that the region set region, so for example, the estrogen receptor binding region, has with the ATAC-seq peak region. And so we can take that into account when we're calculating the score for the region set. But that's all described in the documentation. So really, it's just a similar concept. So basically, now that we've got those scores, we can see a similar outcome for the chromatin accessibility as we saw for DNA methylation. It appears that in estrogen receptor and GATA3 binding regions, there's pretty high correlation between the chromatin accessibility and the estrogen receptor status, whereas there's not really in the ATF3 and NRF1 region sets. And so that's what we're looking at in the ER status column, is the COCO score for each region set for that target variable. And so I won't go through the permutations since it's essentially the same thing that we already saw. But basically, if you want to look at the various scoring methods that are available for region-based data, you can look in the documentation for the aggregate signal GR list function. But basically here we're using the default, and that's going to see what type of data you have, whether it's single base resolution or whether it's region-based data. And that's going to pick an appropriate scoring metric for you. All right, so let's just go through and let's take a look at the unsupervised example, just so you can see what it looks like with the ATAC-seq data as well. And so similar to our previous analysis with the DNA methylation data, we're going to do PCA on our ATAC-seq data. And then we're going to take the principal component scores as our target variables. And so here, just looking at the plots, if you look at the sample plot, it looks like maybe there's some kind of division along principal point 1. And so based on what we know, we can kind of guess that that's estrogen receptor. But assuming we didn't know, that's where cocoa would really come in handy. And so let's look at the first 4 principal components, which is kind of an arbitrary choice. But you could choose that based on the percent of the variation as explained by those principal components. And then let's go ahead and score our region sets. And so here we see that the basically the DNA methylation in estrogen receptor and GATA3 region sets has very high correlation with PC1, less so with the other principal components. And so you might notice that actually for PC2, NRF1 and ATF3 have higher cocoa scores. So I believe that's just because we're only running the PCA on a subset of the data. And we've, for the package, we've subset the data down to only the regions that overlap with estrogen receptor, GATA3, ATF3, and NRF1. So that's why we see a lot of variation there. But if you're running it on basically the whole genome scale ATAC-seq data, you wouldn't see such high correlations for ATF3 and NRF1. All right, now if we go ahead and color the PCA plot by the estrogen receptor status, we can confirm what we had previously thought. That the first principal component is highly related to ER status. It does appear that there's some outliers, but apparently those ER positive samples that group with the ER negative samples must have a similar chromatin state to the ER negative samples, despite being called ER positive. All right, so I'll just go ahead and visualize a little bit of the raw data to help you see that it's very similar to the DNA methylation data. So let's look at those meta-region profiles, since I find that those can be very helpful when you're doing exploratory data analysis. So once again, we are taking our region sets and we're expanding them to be 14 kb centered on our sites of interest. And then we're aggregating the feature scores, which would be the correlation of each peak region with the principal component scores. And we're going to aggregate that into a meta-region profile. So once again, we're just doing some normalization here for the sake of visualizing this. If you just give me one moment. All right, so now we can take a look at those plots. And basically you'll see something similar to what we saw with DNA methylation data. that specifically centered on the sites of interest. So here, GATA3, but we can also look at estrogen receptor, etc. We would see something similar for estrogen receptor. But for GATA3, you can see that the chromatin accessibility is very correlated with PC1 score, specifically at the GATA3 binding sites, but not as much at the surrounding genome. And we can see that that peak is absent for some of the other principal components. So that will conclude the ATAC-seq section of our vignette. I'm not going to cover these because we already covered them for DNA methylation. But now we're going to move on to basically some tips for using COCO effectively. But first, I'll just make sure we get any questions out of the way.
Yeah, so I saw a question that was asking about the typical correlation we see. For estrogen receptor, the correlation in both the ATAC-seq and the DNA methylation data was actually really high, so even above 0.5 for the average correlation for the whole region set. I would say most of the time you probably see lower than that, but it just depends on the biology of whatever systems you're looking at.
There is one more question in the poll. The range of samples that the method is suitable for, so is it suitable for studies with a low number of samples as well?
So I think that if I had a low number of samples, I would use something like PCA, which can really pick up strong signals of covariation across the genome. So I'd probably use something like that instead of maybe a supervised analysis. But we haven't tested that specifically, but I would think that it might work in that situation. Let's see, any other questions, Charlotte?
No, not in the poll. So just to say we have like 3 minutes left, and I see that's more or less what you have on your video too. But there doesn't seem to be any other questions in the chat right now.
Okay, yeah, I'll go ahead and start it. And also, if anyone has any further questions, feel free to just send me a message, and I'm glad to talk to you about our package later on in the conference.
Common question people have is, where do they get their region sets to run this? So there are many sources of region sets that are publicly available. For example, the ENCODE project, Roadmap Epigenomics, various databases like Systrome database, etc. And so we've compiled some of those together into a database, which you can find, there's links, I believe, in the vignette, and in the documentation of the package. But you can find that, for example, in the LOLA package, which has an easy interface to those region sets. So you're also not limited to public region sets. You can also create your own if you want to make custom region sets that might be more relevant. So for instance, you might want to find all the chromatin accessibility regions that are open only in a certain disease. And then you might want to run that through another set of samples and see whether it's relevant. So another question is, are you limited to just PCA, or can you use other dimensionality reduction techniques? And the answer is that COCO is a very generic method. So essentially all you need is a target variable, and you can get that from anywhere. Anywhere that makes sense, of course. But you could use, for instance, another dimensionality reduction method, and you could use those latent factors as the target variables and use a very similar workflow to what we already did. And so I'd just like to finish up. I'd like to thank you for attending. And I'd also like to thank my advisor, Nathan Sheffield, the bioconductor team, of course, and funding sources for various members that contributed to this. All right, so thank you. And now I can answer any further questions that you might have.
Yeah, so that is the extent of our workshop. And as mentioned, I'm glad to answer any questions here or afterwards in the course of the conference. And thank you.
Thanks so much, John.